Our listeners note, Sydney and I are not medical professionals, so we recommend that you do not take our conversation as medical advice and instead talk to your healthcare provider. With that being said, on with the show. When you hear about challenging environments, you likely first think of areas of overwhelming heat or sheer cold, like the deserts or Arctic tundra. However, stressful environments can be found within our very bodies, such as the creation of low oxygen or hypoxic microenvironments where tumors develop. As cancerous cells multiply, they consume oxygen at a rate faster than a body can locally supply it. A new study suggests that this hypoxic environment increases the number and migration of cancerous cells by increasing receptors for a specific neurohormone called neuropeptide Y. Open your ears and mind, and let's chat about that. Welcome to GriffinCast, a podcast where we casually chat about science coming out of the College of Biological Science at the University of Guelph and how their work can affect lives around the world. I'm your host, Michael Lim. With me today is special guest and PhD student Sydney Pascetta, and we'll be chatting about her recent work on neuropeptide Y, or NPY, and its receptors in breast tumors, and how this work can help with treating cancerous cells. Welcome, Sydney.
Hi.
To orient yourself to our guests, and I guess to get you more comfortable, how would you describe your research and or the work done in the UNIAC lab in general?
So our research focuses on low oxygen environments and how they affect cancer progression. So if you imagine a tumor, a solid tumor, it actually does a really bad job at recruiting new vasculature. So as you progress into the center of the tumor, those cells become more and more hypoxic or have less and less oxygen available to them. And traditionally, these would maybe be thought of as dead or non-functional cells, but they're typically the most aggressive cells in the tumor microenvironment. So we're trying to understand what makes these cells unique in that environment, and maybe we could use more targeted therapies towards those cells and not something as generally harmful as chemo and stuff like that.
I guess it's a good point. When you think of hypoxic cells and cells needing oxygen to develop and grow, you expect that, if there's no oxygen, they shouldn't be able to multiply so quickly.
Yeah, but they're actually very aggressive. They're very metastatic. They have a lot of migratory and proliferative capabilities. So that's interesting because it's unique, and hopefully it's not found in the rest of your body. I mean, the rest of your body sits at what we term physioxia. So normal oxygen in the air, ambient oxygen, is 21%, but most of your tissues are significantly lower than that, and the range is usually from 2 to 8%, depending on the tissues, depending on how far away it is from your blood vessels, all that kind of stuff. Obviously, the lungs are the most oxygenated, but your brain is actually really low. So it just depends on the area of the body.
So why did you decide to go into this research? Was there a particular moment you're like, oh yes, I'm going to study cancer cells and hypoxia?
No, actually, I really just liked Jim as an advisor. I went to Queen's for my undergrad, and then I was looking at a few different universities. I was looking at U of T and Guelph, and I met with Jim, and I really liked him. My research in my undergrad was on plant immune systems, so completely different. And I also worked on gut polysaccharides for a summer too. So I've just done a bunch of different things, and I just really liked the lab environment more so than anything.
It's good to try a bunch of different things. Otherwise, how would you know what, you want to do?
Yeah, exactly.
So speaking of research, you recently published a study titled Expression of Hypoxia-Inducible Factor-Dependent Neuropeptide Y Receptors, Y1 and Y5, Sensitizes Hypoxic Cells to Neuropeptide Y Stimulation. So to help kind of guide our audience through that, can you briefly describe what neuropeptide Y does in the body, and what's so special about Y1 and Y5 receptors specifically?
Well, neuropeptide Y is the most abundant neuropeptide in the nervous system. So it has a bunch of different functions, one of which the most classically explored function is actually feeding behavior and eating regulation and stuff like that. But it also acts in vasoconstriction, cellular proliferation, and a bunch of other developmental processes. So the reason why we're actually looking at Y1 and Y5 specifically is because they're the most ubiquitously expressed amongst breast cancer cells specifically. So there's a whole family. There's 6 of them, one of which is non-functional in humans, and then the other ones just aren't really highly expressed in breast cancer.
So the most promising targets, in other words.
Yeah, exactly.
So as part of your research, just to clarify for people, we didn't just
get a whole bunch of cancer patients you're working with breast cancer cell lines. So MCF7 and MDA-MB231. What's the difference between these two types of cell lines, and what are some benefits of working with cell lines versus, grabbing a random cancer patient from the hospital and saying, "Hi, do you want to be part of my study?"
Well, actually, these cell lines are derived from humans. So they all originated from one donor human at some point in time, but they are immortalized cell lines. So we're able to work with them in the lab continuously by doing something that we call passaging them. So we're able to keep them alive. The difference between the two cell lines that we chose is that the MCF7s are ER/PR positive, so estrogen receptor, progesterone receptor positive.
Okay. Yeah.
And MDAs are triple negative. If you've heard that kind of buzzword for breast cancer, triple negative breast cancer is typically viewed as like a very aggressive metastatic form of breast cancer. So we chose two different cell lines in order to understand if the phenomena that we're observing is specific to one kind of like cellular microenvironment, or if it can be kind of generalized across different breast cancer cell lines.
So the different cell lines you used in your study responded to hypoxia and DMOG, which is a chemical used to mimic hypoxia, at different timescales. So you mentioned briefly that these are coming from different individuals with different types of breast cancer, like the triple negative, for example. So what do you think caused these differences in time between your two cell lines?
Honestly, that's really hard to say. The cell lines themselves are quite different. And like I said, the triple negative ones, the MDAs that we were talking about, are typically viewed to be more metastatic and aggressive and have like a completely different phenotype than the MCF7. They're quite different cell lines in general, so they have different signaling pathways that are activated within each of them, generally speaking. So the difference is not surprising, and you would expect there to be like differences amongst them.
Stimulations of the receptors Y1 and Y5 generally increase mitogen-activated protein kinase, or MAPK, signaling in the different cell lines when exposed to hypoxic conditions. So considering the roles of signaling in gene transcription and translation within cells, for our more lay audience, using your DNA to make necessary proteins and function in the body, what does increased signaling mean for cancerous cells?
So MAPK signaling is a really classically explored signaling pathway that a lot of different people have looked at for a lot of different reasons, but its primary role is important for developmental processes. So it's often activated like during embryogenesis and growth and development and all of those really important processes. But like I guess you could say the catch-22 about cancer is that cancer is typically a disease of normal developmental processes gone wrong. So they're normally kind of put into overdrive. So proliferation, migration, all of these activities, cellular activities, are actually normal and good for regular developmental processes. But when these processes become hijacked and they're happening at a much more frequent rate than they should be, or maybe there's checkpoint proteins that aren't functioning properly, so they can't kind of rein in these processes, that's when cancer kind of develops. So by looking at like this pathway, we're just basically saying, can we inhibit or accelerate these different downstream effects? So the MAPK assay that we did at the beginning was, can we actually affect directly this pathway? And then what is the output of that? So that was like the proliferation and migration assays that were done later on in the paper.
Oh, okay. That makes sense. It kind of leads into like the next question, in that obviously, like you mentioned before, NPY or neuropeptide Y, and obviously its receptors and MAPK signaling is very important in terms of regular function within the body and within regular cells. So if cancer treatments reduce the ability for neuropeptide Y receptors to normally function because you're trying to suppress this overactivity, what would this mean for non-cancer cells within the body?
So we wouldn't really look at it from that type of perspective. It would be more in terms of a specialized therapy instead of a chemotherapeutic. So there's a whole new field of personalized kind of medicine and therapy for cancer that's exploring different ways that we can do pointed drug delivery instead of irradiating someone with like a chemotherapeutic or radiation therapy or stuff like that. So there are some labs out there that are looking at bioreductive prodrugs. So basically, when they get into an environment with a different oxygen concentration, like hypoxia, that drug would become active, or using nanoparticles for delivery, or direct like intratumoral injections. So we would be looking more at that like specified drug delivery format rather than affecting like the entire body.
Yeah, I was about to ask you, like, how do you keep it localized in the body? Because, transport within the body, it can, depending on what type of drug it is, could be passed easily across cells. So I guess it's kind of more of a trying to control for hypoxia and take advantage of the hypoxic nature of cancers.
Yeah, that's what we'd be hoping to do. And that's kind of like the ethos of our lab in general. So like this project is one branch of our lab, but our lab actually looks at very, very distinct projects. So there's one that looks at alternative splicing within the ribosome. There's another one that looks at a process called isgylation, which is kind of more immune system driven in a lot of cases. So yeah, there's like a lot of projects in our lab, but it's mostly understanding the biophysiology of the hypoxic microenvironment and what is, like I was saying before, kind of what is unique to that and how we can maybe target that.
So we kind of mentioned this briefly before, and that you were focusing on Y1 and Y5 because they're highly prevalent within breast cancer cells. And so you focus on those receptors as well as neuropeptide Y itself and look at possible treatments. So how applicable would you say your research is towards studying other cancers and trying to address the receptors there by focusing on Y1 and Y5?
Yeah, so it would just depend on the expression levels of Y1 and Y5 in those other breast cancer and those other types of cancer, rather. Because they would all be unique. And there are other cell lines that do express Y1 and Y5 to varying degrees. It just depends on the context and the person, really. But anything that's highly innervated by the central nervous system will have neuropeptide Y stimulation going on, but it's just like a proportion of expression of these two receptors, maybe in contrast with the other ones, etc.
My brain just kind of casting back to earlier in our conversation of how there's a different level between, a hypoxic environment and like a physiologically relevant hypoxia environment. So when you're raising your cell lines, does that mean that a normal environment is actually hypoxic compared to a regular environment already? Sorry if that's a little confusing, but do you know what I mean?
Yeah, so this is actually a really, really important question in science. And there's a whole project in our lab that focuses on physioxia, that range that I was talking about that's actually biologically significant within your tissues. So most research that's done with cell lines raises those cell lines in incubators at 37 degrees, which is what the internal temperature of your body is. But they don't account for the ambient gases. Yeah. So in an incubator, those cells are raised at 21% because that's what ambient oxygen levels are. So actually what another project in our lab has found is that 21% oxygen range is actually quite stressful for cells. It's not their normal physiologically relevant environment. So it's actually pretty stressful for them. And we have another incubator where we can control what the oxygen concentration is. And in those more biologically relevant oxygen concentrations, cells experience less stress in general, like there's less DNA damage and other processes going on. And then the reason why we choose hypoxia to study is because, again, it's relevant to the tumor microenvironment. So a hypoxic tumor is basically a tumor that's around 1% oxygen or less.
Okay. It's all relative. To a regular person, you're like, oh yes, this is totally normal at 21%. But to your body's cells, it's a hyperoxic environment. You're causing excessive oxidation, causing DNA damage. So obviously something we want to avoid.
Yeah, exactly. But most research is done at this 21%. So the way that we understand basic cellular function is for the most part done at 21%. So our lab is kind of bringing research to the table that shows that maybe this isn't, the most relevant concentration to raise them at in general. And like maybe, I don't want to say that everything we understand about how cells function is at least slightly different from the traditional view of it, but it could be, Do
you think there will ever be a moment where you're 100% sure of the connection between neuropeptide Y and receptors and cancer, where you're like, oh yes, this is the driving factor, we should focus all our work on trying to control its expression?
I don't think we should focus our work on controlling its expression, but the one assay in the paper that was called like chromatin immunoprecipitation basically showed that some hypoxia responsive elements, they're called HIFs, they're transcription factors and they're also translation factors, but in this context, transcription factors, they directly bind Y1 and Y5 in their promoter regions, showing that like it is a hypoxic response to upregulate the transcription of neuropeptide Y1 and 5 in the context of these two cancers that we look at. So yeah, so definitely they are important for playing a role in that environment.
Mm-hmm. So is it kind of more of like a positive feedback loop where obviously you don't want to have an excessively hypoxic environment within your cells, with the fact that because of hypoxia, you get hypoxia-inducible factors binding to your receptors and causing stronger effects. So more and more migration, more and more movement, and possibly causing more cancerous cells and hypoxia environments to develop. Is there a way to stop that feedback loop?
Yeah, so that's what we're trying to understand with our follow-up paper. We are currently working on a follow-up paper right now. And so our follow-up paper is trying to understand if we can downregulate these biophysiological responses like the migration, the proliferation, all that kind of stuff, if we can downregulate it by antagonizing the receptors. So in the first paper, we were basically stimulating the receptors, and we're stimulating them both individually and as a whole, because there was the MPY general agonist, and then there was the Y1 and Y5 specific agonist. So we were like, does hypoxia drive the production of Y1 and Y5 in breast cancer? The answer is yes. How is that happening? Well, it might be through the HIFs directly binding and increasing their transcription. And then can hypoxia and the like receptor specific agonists combine together to further drive like proliferation, migration, and all of those kind of things? And the answer to that was yes. So in our follow-up paper, we're trying to understand if you block those receptors, can you antagonize those responses in any way or maybe slow them down?
So developing new treatment is what you're saying.
Yeah, kind of. Yeah.
In general, I think cancer research can be relatively difficult to complete and kind of sell the importance of working on specific, say, cell lines versus working with individuals. And even like we just discussed earlier, the importance of how you're going to hold those cell lines in terms of temperature and dissolved oxygen levels. So what is one thing you wish listeners knew or would kind of reconsider how they think about cancer research?
Honestly, I think the most important thing for people to understand is just how expensive research is in general. Like research in the molecular sciences is extremely expensive. All the reagents we have to use, all of the personnel that we need to use, and all that kind of stuff. Our lab, for example, is a really well-funded lab, and we're like a medium to small size lab, and we go through tens of thousands of dollars of things a month, and that's in a normal regular month. And so I obviously am a big proponent of adding money to like science research and funding and all that kind of stuff, but the funding climate in general in science is actually pretty poor. I think less than 10% of grants, like of people that apply to grants, get awarded them, and like the government doesn't really put it as a top priority to fund science and stuff like that. So it's funny because we're actually very small scale here, but what we do every day is quite expensive. So scale that up to like the big corporations and stuff like that. And obviously there's a reason for privatization and stuff. So so I just think it's important to recognize that the more money that goes into funding these projects, the more forward movements we'll make in our understanding of just the world in general, you know?
I'm not sure how often you read about research in the news, but often you read through the comments like, how could it be spending so much money, millions of dollars on this research? Like, well, it's because reagents cost a lot of money. Companies know that. And you know you've no option. You have to buy from them, so they jack up the price. And what can you do? So research is inherently expensive.
It's true. And most like qualified personnel are like very underpaid. I will say most postdocs, research associates are very underpaid, as well as grad students. So I'm just a really big proponent of like understanding like where the money is going, because people are always like, oh, we raised like $1 million for like a breast cancer walk for for research or something like that, but that might fund like a medium lab for like maybe a year, and when you put it into that kind of context, it sounds like so much money, but then when you break it down, it's like it goes very fast. So yeah, I think funding the sciences is just like super, super important. All areas of the sciences, not just like biology or molecular biology or anything like that, but all areas of science deserve more funding, I think.
So a question I like to ask all my guests, because obviously things don't always go as planned. So if you could go back in time and change one thing about your study, what would it be and why?
I don't know, because we took a long time to publish this. Like, I've been working on it for like, I'm entering my 5th year now. It was started before I came to the lab by a previous postdoc. And we did like 6 months of revisions. So I feel like there was like a lot of thought put into the project. It came off the backs of that postdoc's previous postdoc. So yeah, I don't know. I don't know if I would really change much. I just feel like we're exploring and trying to understand what's going on more so than having like an end goal, really. We don't have like an end goal or a timeline. So the follow-up paper is more exploration of understanding how these things are working. And yeah.
So that's it for me. There's going to be a couple questions I've taken from social media. And our very first question is, at what age should I be more wary of getting breast cancer? Does signaling activity change with age? Does it become more abnormal, for example, and contribute to becoming a more breast cancer risk?
Yeah, so signaling does change with age, and that's why cancer is often associated with it as a disease of aging. There's a much higher incidence amongst those who are elderly or aging in general. And the reason for that is just because those normal biological processes that are happening at all times in our cells are hijacked, are not working functionally, are working abnormally, and that's why cancer develops in the first place. So we have cancer cells in our bodies at all times. Our immune system is just really good at recognizing them and getting rid of them. But as you age, your immune system becomes more preoccupied or maybe less robust than it used to be because you have a lot of other things going on. And some of those cells kind of like escape notice sometimes. And then that's when they can develop into, a mass of cells. And then all of those cells kind of develop into a tumor and et cetera. So yeah, definitely cancer is a disease of aging. It's also sometimes there are genetic associations as well, like whether your one protein is maybe prone to mutations that would make it maybe non-functional or hyperfunctional or whatever. But I don't think there's like a specific age. I think, lifestyle factors also play a role. So just like maintaining a healthy lifestyle, and there's a lot of research that also shows like your stress levels and mental health also play a role in the development of disease states and stuff like that. So I think just generally leading a healthier lifestyle and stuff like that is definitely beneficial.
I actually never really thought about it before, that probably everyone has cancerous cells in their body. And, like, obviously, just the sheer number of cells in your body, some of them will be cancerous, but it's a little sobering.
Yeah, no, it's true. It's a lot of people maybe don't realize it, and and there could be like whatever unforeseen circumstance that maybe your immune system just doesn't detect whatever's going on in that exact moment. But take your vitamins, your multivites.
Eat healthy, exercise, get enough sleep, all that.
It's important. Yep.
So our next question is, are there other symptoms of breast cancer I should be aware of other than a lump? Do you think that it'll be easier to measure neuropeptide Y and receptor levels in the future as a symptom of breast cancer? So
actually, currently, the neuropeptide Y family is used as an imaging tool. So yeah, So they basically attach molecules that could be imaged and then kind of send them through and then see how much you have in your breast cancer. Usually you're diagnosed with breast cancer at that time. But yeah, it is used as a diagnostic tool currently. I don't know if like sheer levels would maybe ever be used as a pure diagnostic tool, but they are definitely used in imaging and stuff like that. So yeah.
I guess the problem is that, like you mentioned before, it can vary between individuals, depending on your lifestyle as well as your
Yeah.
genetics. So unless you have a really good baseline, so you're like measuring it every year, your entire life, leading up to one day you have a really high spike. You're like, oh, that's
Yeah.
weird. Should probably look into that.
Exactly. And as part of our follow-up paper, we do take resections from cancer patients and normal patients as well, and kind of compare them. And they do tend to have more higher expression of Y1 and Y5, but that's TBD in the second paper. But yeah.
You're holding secrets back from us, is what you're saying. And so our last question we have, do you think your work will replace other forms of cancer treatment like chemotherapy? How long do you think it will be before it's more widely used or talked about?
I mean, I think the hope would be that we don't have to use something as aggressive as like chemo and stuff like that, because that just basically kills all the cells in your body and hopes that you irradiate like whatever is the cancerous hub, right? But the problem with cancer, of course, if the problem with cancer were just a tumor, we would be able to surgically remove it and every patient walk free and be fine, right? It's those single cells that kind of migrate away from the tumor in microenvironment and then establish themselves in other tissues. So that word like metastasis is kind of like a huge negative buzzword, I suppose you could say, for cancer. So if we can like understand how those cells in particular are functioning and maybe target those a bit more effectively, that would be really promising for research in general, and like cancer research in general. So yeah, I think we would hope to just generally move away from things that would, make people otherwise very sick and frail and stuff like that.
All right. So before we end, if there's someone listening who say maybe in undergrad or in high school and is thinking about what they want to do you recommend they also go into cancer research, or what words of encouragement or advice would you have for those people?
I think as scientists, we're all just like curious. I mean, I know for myself, like most of my friends who are grad students don't necessarily care about the specific thing that they research. I mean, some do, but I would say for the most part, most of us don't. I think we just enjoy the curiosity of science. And, for one moment in time, you're the only person in the world that knows that result or knows that like thing. It's true.
You're the foremost expert for, however long it takes you to publish the paper.
That, but like even like as you're reading, an assay or something like that, or waiting for results to come out, you are the only person in the world that knows what those results are and what they might mean. So I feel like that's like, that a really exciting, intangible experience with science. So yeah, like I don't think most of us really, I don't want to say we don't care. That's not the word I'm trying to use, but like we're just like,
it's one component.
Yeah, we're just like excited to understand things about the world, and it doesn't really matter what those things are.
And so with that, we come to the end of today's podcast. A big thanks again to our guest, Sydney Pascetta, for joining us today. Griffin Cast is brought to you by your host, me, Michael Lim, with editing assistance from Ian Smith. If you're hungry to learn more about different science topics, please check out Scribe Research Highlights. That's SCRIBE, Scribe Research Highlights on the University of Guelph website at uoguelph.ca. Or you can follow us on social media at UofGCBS. You can find us on Instagram, Twitter, and Facebook. Music in the podcast comes from Uppbeat. There'll be details in the show notes as always. And until next time, stay curious.